Recording?
Yeah.
Cool.
Let's go.
All right. I think at this point we should pause, pick one of these things, do a quick deep dive to review how all the pieces connect together end to end.
Okay.
And then we'll get back to introducing new things. So let's say. How about this guy with the feedback loop going on?
Okay.
This one's the positive feedback loop.
Yeah.
All right. So here we were talking about how a positive feedback loop can make the thing bistable, so it can be used as a bit of memory.
Yep.
And then you expect that's like a thing that's potentially useful for selection pressures to start using as a bit of memory, so that the cell can be keeping information over longer time spans.
Yeah.
How did that build up on top of everything else? Well, we needed something to evolve, first of all, in order for this evolutionary argument to make sense at all. That came from these sort of modular molecules made of interchangeable pieces that came out of autocatalytic cycles, sort of sealed inside of these membranes. All of those pieces came along in some order, and we're not necessarily sure which. That, in turn, we built up on top of this idea of objects, of things that sort of these patterns that are conserved over time, and they have these sort of dual quantities that are conserved over space that act sort of like a position or an orientation or something like that.
Yep.
So, for instance, we get many molecules of species A. This indicates that we have this pattern that's conserved over space. Each of them has associated with it a sort of position, orientation, and some other degrees of freedom that tell us sort of how that pattern is embedded in this particular instance of molecule A. And a particular molecule A may similarly stick around over time, and we can say sort of how that's embedded in place. And then that, in turn, was built on top of symmetry and locality, before that, symmetry and locality, and then chaos sort of wipes out all the other information that's not conserved, which is what makes these quantities conserved over time and over space sort of natural things to look at on top of this low-level physics.
Okay. This still feels a little bit like here is a frame for thinking about the universe.
Yep.
Now, let's talk about how life evolved.
Yes. Yes. So a little bit of preview of where this is going. We have some more biology to do. Couple more steps before multicellularity, then we'll talk about multicellularity for a bit. Then we'll start to talk about higher level minds like animals and humans.
Okay.
That's when we're going to come back to things like objects and why those would be sorts of concepts that humans and animals also end up using.
Okay.
Cool?
Yeah.
All right. So we just finished talking about things serving as informational signals, turning into more complex wiring diagrams, feedback, bistability, memory, those things. Next up is going to be consistent trade-offs. So we very briefly started to touch on this sort of thing over here, talking about we have two molecules here, and how they start to specialize in different roles.
Yep.
This one specialized in being the blueprint, and the other one specialized in whatever functional stuff.
Yep.
More generally, we expect to see lots of different molecules that are specializing in different roles. Once that starts to happen, we expect selection pressure for consistent trade-offs. This comes from your classic economic argument, you've got the two people and they're two separate islands and they can both produce apples and bananas, and they can produce apples and bananas at different rates, different trade-offs, so they can sort of jointly produce more apples and bananas by one of them specializing in apple production and the other one specializing in banana production, right?
Yeah.
All that jazz. Similar thing here, if you've got different reaction pathways can produce the same molecules, for instance. A cell can use either of two different food sources to produce the same things, or it can use different intermediates to do the same things. There are places where like the cell can substitute one thing for another, right?
Okay.
And then what we expect to see is selection pressure for Pareto-optimal usage of all these resources. So, for instance, if let's say a cell has two energy sources, right? Let's say it's got glucose G and lactose M, right?
Okay.
And it also has two molecules it needs to build. I don't know what molecules it need to build. Let's just call them A and B.
All right.
And like it can use glucose to power some process that produces A, or it can use lactose to power some process that produces A. And because it's sometimes in a glucose environment, sometimes in a lactose environment, it's been selected to be able to produce A out of either of them.
Okay.
And similarly for B, it could use either glucose or lactose to produce B. But maybe this thing is in a point in its evolution where it's not yet like near perfectly energy efficient with every energy source and every sink. So maybe it has different efficiencies here. So maybe it needs like 0.5 units of glucose to produce an A, and 12 to produce a B, or only 6 units of lactose to produce a B, or 0.4 units of lactose to produce an A. And now we've got this thing where when it has both of them available, it makes sense to specialize, right? The lactose is relatively more efficient at producing B. The ratio of 6 to 0.4 is smaller than the ratio of 12 to 0.5, right? So it makes sense to sort of specialize your lactose for producing B, specialize your glucose for producing A, insofar as you have both of those things available.
Yeah, although if you're in environments where you are heavy on glucose or heavy on lactose,
then you need to swap back to the other, right?
yeah.
So this the part where you like swap based on how abundant the glucose and lactose is, what this is indicating is that you want there to be a sort of consistent pricing of glucose versus lactose, sort of consistent trade-off ratio between the two.
Yeah.
So if you have a glucose to lactose ratio of, let's say, 1 to 3, then you want to use lactose for, and that's like your environment state right now. Your current environment has 1 to 3 ratio. Then you want to use lactose for all the things where like your lactose to glucose ratio is better, your trade-off ratio is better than 1 to 3. So like if I can get more than 3 times as many Bs for using lactose versus the same number of A's instead of glucose, then I want to use my lactose for Bs.
Yeah.
Vice versa, right? So there's this incentive pressure to have what an economist would say are consistent internal prices, consistent internal trade-off ratios.
Or there are arbitrage opportunities.
Exactly. Where arbitrage opportunity here means there's an opportunity to get some sort of Pareto improvement in these like molecules that are being produced, which ultimately we expect to cash out to an improvement in the reproduction rate.
Okay.
So we naturally end up with a sort of consistency pressure that's going to give us at least approximately consistent internal prices.
Okay.
Or consistent internal trade-off rates. All right.
So then our cells are coherent.
In that sense, yes. Where did my blue go? There. And now note that this is a thing which like we only expect to hold approximately if there's like as long as things are close, there's going to be quantitatively small selection pressure for it to get more precise. So that's should be an approximation. All right, 2 more topics before we get to multicellularity. Next one is mobility. So far, this cell is just stationary, right? Turns out bacteria can swim around.
Yep. All right.
Yeah, so it's got this little flagellum that it spins in a circle and sort of powers it along. How the heck did that come about the first time? Presumably, at some point, there was some sort of molecule that could burn energy to move. That's the sort of thing which maybe came along by accident. Maybe it evolved out of some other molecule that was burning energy for something else, and then just lost all the other functional parts except the movie bit. But at some point, there was a molecule that could burn energy to move itself around, and then that could give the cell a little bit of a kick in a direction, and then eventually that evolves into something that's more and more specialized in moving the cell.
Yeah.
Now, just a cell moving around by itself is already something that you'd expect to have a pretty large selective advantage in an environment where cells are mostly not mobile, because it means they can spread out a lot faster. Just like with plants, they have a large selective advantage if they have some mechanism for dispersing their seeds over a wider area, right? Because we have fruits that are attracting birds, or we have dandelions that are supposed to blow their seeds around on the wind, or what have you. Same thing here. Once this cell gains the ability to start powering along, that's an immediate selective advantage just because it can spread out more and end up in more places. But random motion's only going to get you so far. Pretty quickly, you expect that to start coupling to all this other jazz, right?
Yep.
So
pretty soon we mean some millions of years later.
Presumably, yeah, exactly.
I'm actually not that clear on the time scale. I think
it would be somewhere between millions and hundreds of millions, I think.
Okay.
Yeah, I haven't read these things carefully enough recently to be confident in that.
Yeah.
But yeah, pretty quickly that we expect that to end up coupled into all this other stuff. You get some sort of control system. In the case of E. coli, there's basically 2 states of the flagellum. It can be in the go forward state or the flail around state.
Okay.
And what it'll do is it has a little internal circuit that keeps track of how quickly local concentrations are changing.
Yep.
As it's swimming along.
Concentrations of
Sugar, for instance.
Okay.
It can be more than one different thing, but sugar is the classic example. So if it detects that sugar concentrations are getting higher as it swims along, using something along these lines, then it'll keep swimming along, keep spinning in the direction that makes it go forward. If it detects that sugar concentrations are going down, then it'll flail around, end up facing in a random direction, and start swimming that way.
Yeah.
And at this point, we can see how we could cache all of that out into lower level pieces. All right, last piece before multicellularity is immune system. So somewhere along the way here, we start getting parasites, viruses, that sort of thing. Probably a lot of them in the early days. And then you need there's going to be strong evolutionary pressure for something that'll defend against those sorts of parasites, right? Thus we get things like, for instance, CRISPR, the proteins that we built CRISPR out of, that detect, they will bind to the kinds of RNA or DNA sequences that viruses are typically made out of, and just slice those things up into little pieces.
Okay.
That's your sort of simplest possible immune system. Once we get to multicellular organisms, of course, we got more complex immune systems with specialized cells. But simplest example would be something that, for instance, just binds to double-stranded DNA. Which will come later, but binds to double-stranded DNA, slices it up, Because if you see double-stranded DNA outside of nucleus, something's wrong.
Okay.
All right, that's everything before multicellularity.
Everything.
Well, it's everything in my notes.
All right.
We don't know the order in which these things happen. So.
Yeah. And I guess we won't until after the singularity. Maybe not even then.
Most likely, yeah. All right. For multicellularity, we're going to take a small step backwards first and talk about self-assembly in general. So, like, how do things self-assemble? Why does that happen? Shot of this. So a few kinds of things that self-assemble. First one would be something like a crystal. So you've got a bunch of identical atoms or molecules, and their like energy minimizing structure is something regular like this, right?
Okay.
Yeah, pretty straightforward. Next would be something that has a sort of more interesting three-dimensional shape. So in the case of a crystal, you just have like this sort of tiling that's filling a whole 3D space. But you can also get molecules which naturally self-assemble into, for instance, a tube shape. So you'll get these little dudes that sort of like spiral around, and then as they spiral around, once they get back to here, they'll start sort of going on top of each other. So this thing is sort of spiraling out of the board like that.
Yep.
And it'll naturally assemble into a tube.
All right.
And for the same reasons that things like self-assemble into a crystal, it's just like a different shape of thing.
Are the walls of the two bound to each other, or they're just sitting next to each other?
Yeah, so what'll happen is as the spiral goes around, this layer of the spiral will bind to the next one.
Okay.
So you'll end up with the thing like pretty tightly structured there. Then a third thing which sometimes happens is you get things that self-assemble into a 2D shape. So very much like the crystal, except it'll self-assemble into like a thin film rather than a whole crystal.
Okay.
I'm emphasizing these right now because these are sort of things that are natural shapes for stuff to self-assemble into. So they end up being natural shapes for things to be built out of inside of biological systems. So for instance, on the tubes front, there are a few different kinds of like tube-shaped things that cells will produce. Actin is a good example. Collagen is another good example, and these are like sort of the primary support structures inside a cell that will give it a shape.
Okay.
So, for instance, in muscles, you've got these long actin fibers. And these are like sort of the ropes that get pulled on.
Yeah.
Or in like your hair, right? You've got these long fibers, or in your skin, you've got keratin fibers. All of these like fibrous things are ultimately coming from these nanotubes of whichever things are self-assembling into a tube, like collagen or actin or whatever. Yep. At another level, or another example of a tube would be, for instance, you've got things like your blood vessels or your lungs. Similar like dynamics there. You can get things to pretty easily self-assemble into tubes at the level of cells, right? It's just at a larger scale.
Yep.
Then when we think about things like planes, typical thing would be a surface that like separates any like compartments inside of your body. Cells will form these 2D planes basically that sort of separate one side from the other.
Okay.
And those are all standard shapes that like stuff gets built out of in a multicellular organism.
Yep.
Yeah.
Or a single cell organism.
Or a single cell organism, yeah, inside of it. Right. So getting back to multicellularity itself, where does it start? Most likely, the very first multicellular organism is just going to be a sticky ball. So
Okay.
You've got like some cells. At some point, they have a mutation that produces something on their surface which is going to stick to other cells of the same type, and then you end up with this sticky ball of cells. Why would that be selected for?
Seems not good for the cells on the inside.
Partly true, yeah. But remember, these are all clonal cells.
Yeah.
So, like, the selection pressure isn't necessarily acting on different cells. It's acting on
Okay.
the like clonal population as a whole. So if there's a mutation that lets something get closer to the surface, that'll be like evolutionarily selected for it. But other than that, the fact that there's some in the middle that are dying off just doesn't really matter that much.
Okay. So why would it be selected for? Well, it's bigger.
All right.
There's more surface area.
Ironically, the opposite.
Oh yeah, I guess less surface area.
Less surface area per
unit volume. Yeah.
Yep.
Um,
However, that means that if these things benefit from less surface area per unit volume, that's good. So, for instance, it would probably be a little bit hotter.
Yeah.
Although at this scale, debatable whether that would matter at all.
So I think the main thing is it's just hard to. Yeah. Are there advantages to scale? Why does the population do better if they're stuck together?
It could be that the cell. This is a cell type which is typically modifying its environment in some way to like make it more suitable for itself.
Okay.
And then if there's a bunch of them close together, then that's cheaper to do.
Yeah. Okay.
It could be
It's just
that.
economies of scale, basically.
Yep, so that's a possibility. It could be that these are cells which benefit from sticking to things that move around, like big things.
Okay.
Or maybe they benefit from sticking to a
Things that
particular
move
place.
What moves around?
Rocks,
Okay.
they blow around sometimes.
And then they also stick to themselves as a spandrel, basically.
Yep, that would be a possibility. Maybe they just like sticking to walls.
Okay.
Like sometimes a wall is a nice place to hang out. Yeah, so we don't really know like what specifically would have made the first sticky ball of cells want to be a sticky ball of cells, but probably the starting point was a sticky ball of cells because like it's a very simple thing to evolve.
Yep.
All right, now where multicellularity first starts to get interesting is when these things start to specialize. So you've got your sticky ball of cells, and now maybe some of these cells start specializing in different roles. One obvious possibility is probably the cells in the middle should be doing different things than the cells on edges.
Yeah.
So don't know what that would be, but like maybe cells on the edges are specializing in like absorbing stuff from the environment and producing like basic intermediates that they're then going to spit out to be absorbed by things more towards the middle-ish. That would be a possibility. And then that specialization would be affected through like wiring similar to what we discussed earlier. A cell that sort of detects that it's like seeing the sort of external concentrations that tend to be in the middle of this ball will then go do things that make sense for a cell in the middle of this ball. That's how the selection pressures would be pushing it.
Okay.
So we've got like some specialization between stuff more in the middle or stuff more on the outside.
Yep.
We could also imagine other kinds of specializations, maybe even separate from the inside-outside distinction. It makes sense for some of these cells to just randomly specialize in one thing and some of the cells to randomly specialize in another thing. And this is a thing that we see in developmental patterning all the time, is like at certain stages, cells will randomly decide, I'm going to specialize in this, I'm going to specialize in that. They do that.
Okay.
So that's another possibility. And now this is similar to earlier we were talking about, like you could have internal compartments and then like those specializing things. Now we've got the same thing, but across cells. That's great. Much simpler in some ways to specialize across cells because like you don't need to make whole internal compartments for it. Now we start to get into sort of basic patterning. The sort of simplest starting points are the things that happen very early on in embryogenesis. In particular, you'll get a little ball of cells, and it'll lay out a chemical axis. So there will be some sort of chemical gradient from one end to the other.
Yep.
And this is how you get this serves as sort of backbone for a body plan, right? So like this end might become a head, this end might become a tail.
Okay.
Then slightly more complicated, or actually, let me back up. Note that we're no longer in a single cell here. So the way this has to happen is some cells up here are going to be like producing the chemical. Some cells down here are going to be maybe eating the chemical or just not producing it.
Yep.
And there needs to be some sort of feedback loop here that's making cells at one end produce more, cells at one end produce less, such that you get like a directional gradient over the thing.
Okay.
The thing to Google here, if you want to read about like what sorts of reactions will do this, is reaction diffusion systems.
Okay.
These are like how you get particular chemical patterns like distributed through space that can be used in this way. So at some point some circuitry happens to evolve or happens to have already evolved such that you naturally end up with gradients, some chemical gradient from end to end of the cell, and that gets used as an informational signal for specialization. So similar to like the inside-outside thing, they have stuff at one end specialize in one thing, stuff at the other end specialize in the other thing.
Okay.
Then after that, you start to get a full body axis system. So the like end-to-end thing would be the simplest circuit. The next more complicated thing would be you get a second chemical gradient orthogonal to that one.
So when you say chemical gradient, you mean some cells are emitting a chemical, and so there's just higher concentrations of it at one end than at the other.
Yep, exactly.
And it's diffusing through the cells?
Yep.
Is passing through the membranes?
Is not necessarily diffusing in and out of the cells. It is diffusing across the population of cells. So in the extracellular space.
Okay. So the fusion is happening between the cells.
Yep. So like sort of oversimplified picture to have in your head is you've got a bunch of cells with some space between them. Some of them they're all going to be like producing or absorbing this chemical based on some simple rules that are like hard coded by the wiring diagrams. And then the net producers are going to produce a bunch of this chemical. The net absorbers are going to be absorbing a bunch of this chemical, and you'll get the chemical diffusing between the cells, so you get this chemical gradient going from like high to low.
Okay. And what triggered the cell at one end? Just some one cell randomly starts being like, I'm gonna be a net emitter.
Yeah. So my guess, so probably in like the earliest versions of this, that's what happened. And in some places, developmentally, that does happen. So for instance, classic example of this would be in a fruit fly's eye. This isn't axial patterning, but in a fruit fly's eye, some random cells will decide I am going to be like a new epicenter, and then the sort of insect eye grid pattern that you get is like around those cells that decided they're going to be the center of a little chunk.
Okay.
And like what they will do is they'll produce a bunch of this molecule that will suppress the cells around them from producing that molecule.
Yeah.
And that way this one cell gets specialized in being sort of like in the middle. And because it's suppressing things around it, they'll end up reasonably spread out.
Okay.
So that would be an example of how it could happen. However, in embryogenesis, when these are like in a human embryo, for instance, when this pattern is first being established,
Yeah.
it begins with there's a slight electric asymmetry right from the get-go. One end of the cell is slightly positively charged, one end of the original cell is slightly negatively charged.
Okay.
And you end up following that asymmetry and laying out the body plan.
Where does the electrical charge come from?
That part I do not understand myself. So
Okay.
I'm afraid I can't answer that.
Okay. All right. So you have an one axis, and then you have the orthogonal axis.
Yep. So the layout of the orthogonal axis is a more complicated thing because you need like some sort of tricky rules for the cells to follow and like how much of the chemical to produce such that it ends up sort of naturally orthogonal to the other axis.
Okay.
But again, there are rules that produce that, relatively simple rules that produce that.
Yeah.
And then, of course, you get a third axis, because here we are in three-dimensional space. So you get these three axes that lay out the body plan. You get the this way, that way, the this way, that way, and the this way, that way.
Okay.
Yeah, so that's some basics of embryogenesis and body axes.
Yeah.
Next big thing is gas relation. So once you've got those axes laid out, lots of organisms develop a stomach.
Okay.
You may have noticed that topologically you are something of a tube.
Yeah.
That's the tube shape shows up extremely early on in embryogenesis.
Yep.
So your cells will like sort of fold in on themselves, form this tube shape. That's okay. Turns out I can't really draw a tube. I didn't know this before right now, but yeah, this is not getting better. I'm gonna stop. So there's a tube, right? They form this tube shape following the main axis.
Yeah. All right. So we've evolved a sponge.
Basically, yeah.
Or a worm.
Yep. And again, this is all happening through like these reaction diffusion signals are telling these cells like how to specialize. So like which ones are going to sort of become the middle ones, which ones are going to become the outer ones, and then they sort of adjust how strongly they're binding to the cells next to them so that some of them will naturally migrate inwards.
Yeah.
So that's gastrulation. Note that the gastrulation step is sort of it's the membrane thing again. It's like forming a boundary. So you've got like a clear inside and outside, and you've got stuff can pass between the inside and outside, but now it's at a much larger scale.
Things passing from the inside to the outside. You mean the inside and outside of the organism?
Yep. So like originally we were talking about this in terms of like very early life forms. You get a membrane forming. Why does a membrane form? We're not really sure, but like various reasons that it's useful to have a separate inside and outside.
Yep.
Same thing here. Now you've got a through gut or even a non-through gut, even if you just have a sort of cavity in there.
Yeah.
Now you've got an inside and an outside. You can suck food in, digest it in there, have a chemical environment that's very different from what's outside.
Yep.
And the whatever selection argument you have for like a membrane, you have similar selection arguments here. Next up, now we get into like more complicated patterns. We started talking about this before. You have these sort of like shapes that are natural to form with relatively simple rules, things like tubes, surfaces, and bulk crystals, for instance. And all of these things come up once this organism starts patterning itself, right? Maybe it forms some outer layer that's like sort of a plane that's wrapped around the thing, so like this sort of thing. Maybe it forms some internal branching structure to carry nutrients around. All of those are possibilities.
Okay.
Next, so patterning. We've also started touching on pieces of this. We've talked about, we mentioned the reaction diffusion thing. That's sort of the starting point for patterning. We've talked about the fly eye, actually. Apparently that was in my notes. All right, I'll put the picture in with the others. So the fly eye thing, various cells will randomly decide to start expressing this particular molecule.
Yeah.
That'll suppress cells nearby from suppressing it. So if there's two close together, one of them will win out.
Okay.
And if there's like a big space without any, then it will be not suppressed. So like something will tend to decide to start expressing in there.
Yeah. Okay, and so they end up about evenly spaced.
Yep, so you end up with this nice even spacing.
Does that mean that the insect eyes are not like the individual?
Not a perfectly hexagonal grid?
Yeah.
I believe so. It's not a thing I've personally checked, but that is my understanding.
Okay.
So you get these little compartments. Yes, I'm drawing this right. You know what? I'm gonna stop now. Yeah. It's that thing. Then we get like the really cool stuff for segmentation. I wish I had a third color marker. There.
We don't have enough markers really, but I can find some for you.
Yeah, just. No, we don't have a third color marker. All right, I'm gonna draw this in two colors. So there's this. So imagine something like an extreme example would be a centipede, right? You've got like these body segments one after another, right? So in order to lay out the original chemical signals to form something like that, what you want is a periodic chemical concentration. So something where like along the length of the organism early on in development, you get a concentration that is varying like this. right?
Okay.
So how do you get something like that out of a reaction diffusion system? Well, it turns out you use 3 separate components in such a way that their concentrations will naturally be a little bit offset from each other. So quite nailed that. That's about right. So you'll have these things like being produced and diffusing in such a way that, for instance, maybe if you have a high concentration of solid black, then you'll be producing reasonable amount of blue, and then blue will sort of net diffuse this way. You'll end up with a blue pattern that's offset a bit from the black pattern.
Okay.
And then similarly, same thing again with the dotted black versus the blue. You get a thing that's sort of net offset. And then similarly with the black and the dotted, you get a thing that's net offset, so you get this sort of periodic pattern.
Okay, so each thing is enhancing or amplifying the signal behind it and suppressing the signal
ahead of it. And the crazy thing is that it doesn't really, these things don't really know that there's not the like ahead and behind are kind of tricky concepts here. Because like other than the underlying body axis, like the underlying body axis is the only thing that tells you ahead or behind, right?
Yeah.
You just have an idea of where that is.
Okay.
But yeah, it turns out there's like some relatively simple rules that you can use with 3 molecules to get this pattern. And there's like some of the coolest visualizations you'll ever see in a paper. They visualize the concentrations of these molecules in an embryo, and then like track where the segments form of its body, and they like perfectly match up to the like wave pattern there.
Okay.
Yeah. So that's segmentation.
Alright, so basically you make a sinusoidal pattern by having three signals that are amplifying and suppressing each other.
Yep.
Anything that has most animals that have body segmentation don't have equally spaced body segments. So something needs to happen. Maybe this is more complicated than it's worth getting into right now, but something needs to happen to stretch the sinusoidal segments to match the body segments.
Correct. The example we're going to use here is a human spine.
Okay.
So like you've got these segments along your spine, right? The little, dudes sort of stacked one on top of each other, but
Yeah.
they're not all the same.
That's right.
They get sort of smaller as you go down. What does that?
Bigger as you go down.
What was that?
They get bigger as you go down, I think. There's
Well, like right in the tailbone, they're tiny, right? Point is, they vary in size as you go down.
Yeah.
Yeah. So what's responsible for that?
Oh, you're right. Okay. But you do have a pelvis, which is very big, and so I was.
It's a different thing. It's not a spinal.
Yeah, that's right.
Anyway, next thing, Hox genes.
Okay.
So Hox genes, there's this like sequence of genes that's physically laid out one after another on the genome. And if you look at spatially where in the organism they're active, you'll find that if this is the body, at this end of the body, you'll have like all of these genes are active. And then when you're right here, it's like all of those genes are active. When you're right here, all of those genes are active, and so on down the length.
Okay.
So you've got your like main body plan chemical gradient controlling all this originally.
Yeah.
And there's some whole sort of like thresholding style thing going on, or some sort of thing going on where this one is only active if you're like have really high concentration of this thing.
Yep.
Then this one will be active when there's like threshold slightly lower, so on down the line.
Yep.
Those are Hox genes, and they'll control, like, depending on how far along you are, which things are going to develop. So you get, for instance, different size chunks of your spinal column down your back, right?
Okay. So basically, and this is built up of the thing that we covered before, where if there's high concentrations of chemical A, then you put a protein right in front of. I guess you put a protein after each of these actually.
That just like blocks the thing.
Correct. Yeah.
Yeah.
And it blocks it if and only if there's some threshold of the concentration of the chemical that's being diffused from the net emitters at the top.
That's basically the right idea. I don't know for Hox genes specifically whether it's a chemical that blocks it or a chemical that activates it.
Okay.
By like, allowing transcription machinery to like bind and start there. But one of those two.
And we're now talking about modern developed organisms.
Yep.
Is it actually a protein? Is it or that's sitting there on the genome? I am imagining there's more complicated.
You mean the actual activator or repressor that's like
Yeah.
binding here? Yeah, it's a protein that's sitting on the genome. It's binding directly to the DNA.
It's okay. It's not. There's not a gap in the DNA. It's sitting on top of it, and it matches some particular nucleotide sequence.
Yep.
Okay. All right. So there are a bunch of regulators, protein regulators on DNA that is not itself genetic.
That's correct.
But I guess the DNA constructed the.
Ultimately,
all the
the
regulators.
DNA provided the code for constructing them.
Okay. Okay. All of the conditionals that that DNA can execute are non-DNA proteins that are sitting on top of the DNA. Is that right?
Basically,
Or
yes.
binding to it.
Basically, yes. Some of it may be RNA occasionally, but
Yeah.
in modern organisms, generally protein.
Okay. Cool. Okay.
Okay, so to date, like, we definitely don't have a full and comprehensive understanding of everything that goes on in like happening in multicellular organisms, but this is the like nice sampler platter of the things we do have some understanding of. So like typical patterns that come up again and again. You'll see Hox genes are used in lots of different places. The like characteristic pattern here is you've got a sequential pattern on the genome itself that directly maps to a sequential pattern in space.
Yeah.
You've got these segmentation patterns. You've got tubes and surfaces that form naturally. You've got these sorts of patterns where like one cell sort of becomes a little epicenter. And you've got axial patterning. Those are sort of like there may be other building blocks that we don't yet understand, but those are like the building blocks that we understand to date, out of which like things get built.
Okay.
Make sense?
Yeah.
Cool. Next up. So that's mostly about body plans. Next sort of high level thing within multicellular organisms is going to be specialized signal carriers. So neurons, for instance.
Yep.
Probably before we got neurons, we had other kinds of specialized signal carriers.
Yeah, chemical before electrical, I would imagine.
Probably, yeah. Although the the ion channels that like form the basis of neurons did come pretty early. But,
Okay. So maybe
neurons, yeah, but still probably had chemical signals before then. So what would be like the sort of bare minimum signaling function that a cell could specialize into? That's the question right now. So we've got at some stage of evolution, it may still be at the sticky ball stage, it may be at something later than that, not sure. But we've got some sort of cell that's specializing in passing information.
Okay.
What does that look like? Well, probably there are some other cells which already like detect a particular molecule of some sort and will do something in response to that. And then this guy is like, all right, I'm going to produce more of that under certain conditions.
Yeah. Okay. It's just an amplifier.
Mm-hmm. So, very made up example here, for instance. Maybe there are certain molecules in response to which these molecules will like start swimming. Or, sorry, certain molecules in response to which these cells will start swimming. So the whole thing sort of gets pushed this way, right?
Okay.
And now, if say this guy detects something that is correlated with predators,
Yeah.
then it's gonna maybe it starts producing whatever molecules make these guys swim, so that the whole thing swims away. Right?
Yeah.
You could imagine that would be a thing that would be selected for, and also relatively easy to evolve. So that would be sort of the bare minimum starting point. Once you start to get that, you could also imagine these things start chaining. So, for instance, if you've got like multicellular organisms, things got some size to it, sometimes there's going to be some information detectable at this side, and what you want to do is tell the guys at that side to go swim, right? So now, there's selection pressure to get a whole chain of these amplifiers.
Yep.
So this one starts producing a molecule, it triggers that one to produce it, triggers that one to produce it, all the way along until the thing's swimming.
Okay.
And you will want that to be somewhat gated on the body plan. Like,
Somewhat gated?
yes, as in like, you want this guy to be sort of the guy who kicks off this signaling cascade.
Yeah.
And so you might want him to have somewhat different behavior than a guy in the middle who's just passing a signal.
Got it. Okay.
So that you don't accidentally get like stuck in the let's go swim position and never turn off. Right? Yeah. So amplifiers, conditional amplifiers with like different functions, so that they don't sort of get stuck in the on position. That's sort of basic signal passing machinery. Now I've got that. Now there's probably going to be selection pressure to pass that signal both more quickly and more reliably. So if there's selection pressure to pass the signal more quickly and reliably, what sorts of things is that going to select for?
Barriers that, or I guess, I don't know, myelination? Something?
Yeah, that would be qualitatively the sort of thing. At this earliest stage, probably just something like long cells,
Yeah.
right? Or
Okay.
another factor here might be something like they have nice S-shaped response functions, so that you get like a nice binary signal. So like if the signal from this cell is coming in, if it's low, then the signal in the next cell is going to be lower. If it's high, the signal in the next cell is going to be higher. So you get this like nice clean signal that doesn't degrade too much as it like passes along the cells. So that's one thing that will be selected for. And these long cells like carry things a long way quickly. Be another thing that's selected for. And eventually, like where that's going is towards neurons, right?
Yeah.
Like these long cells, they eventually. At some point, they start using not just chemical signals, but electrical charge across their membrane to carry these signals from one end to the other.
The individual neurons have sigmoid-ish activation patterns?
Yes. This is a...
Not Redloose?
No, not Redloose. This is a thing that's really important, for instance, in electrical engineering.
Yeah.
So quick side story here. In the very early days of building electrical computers, a big problem was if you tried to make these things at scale, make things with a whole bunch of elements in them, noise will just wipe out your signal, right?
Yeah.
Because you've got these sort of deeply nested circuits, and every level is adding noise. Shannon's master thesis, same Shannon who invented information theory, his master's thesis was about how to solve this problem for digital circuits. And basically the answer was make your components all have shapes, activation functions like this. So that low signals get lower, high signals get higher at each stage. So now when you introduce a little bit of noise, as long as the signal's still low, that noise will be killed off, it'll be made lower.
Right, okay.
Introduce a little noise that's high, well, that signal's still high, it'll be made higher, and the thing will stay nice and clean as it propagates.
Which is the basic advantage of digital over analog.
Exactly.
Okay.
And so there's very strong selection pressure for that sort of thing to show up, that sort of bi-stability, quote unquote, to show up in anything that's specialized in passing signals over a long distance.
Yeah.
All right, so that got us basically to neurons. Okay, now another thing you might expect to show up at this point is, well, you're not just going to have a line of neurons, right? Possibly you're going to start to have a whole web of these things spreading throughout the organism. And that is what we see in things like jellyfish. They don't have brains, but they have a sort of dispersed network of neurons that are passing a signal around the jellyfish. So if you poke it, it'll be swim appropriately.
Okay. Jellyfish do they have a protospine?
I don't
Or
think
is it
so.
all just
I think it's just a decentralized net.
Okay.
Again, assuming I'm remembering this correctly. So now we've got these things that are basically just information carriers. Natural next thing to happen is they start doing some information processing in addition to just passing the signal along.
Okay.
So even this S-shaped curve is already very basic information processing, right? At each stage is sort of cleaning the signal up a bit.
Yep.
You can imagine pretty quickly, there's going to be neurons that are connected to multiple other neurons, and they're going to have some activation function that's sort of summing the signals, conceptually summing the signals from the different neurons. That sort of thing will just sort of naturally show up here. So you'll naturally be doing some information processing, and so that can naturally be selected for specific circuits, right? Insofar as these patterning signals are able to control the circuit topology of these neurons, there's going to be selection for the information processing to do useful things.
Yeah.
So at that point, we expect to start seeing processing circuitry, basically. In particular, two types of patterns we would expect to be useful and show up. One big one is, again, bi-stability, for the same reasons that we had it at the molecular level. If you have some sort of bistable circuit, then that can act as a bit of memory.
Yep.
And then another one is potentially you start to see some sort of learning, something like Hebbian learning, neurons that wire together, that once you've got this machinery in there, that's a pretty easy thing to evolve insofar as it's useful.
Okay. Why is it useful?
Well, so in the case of neurons that wire together, that's a learning mechanism. If these neurons are sort of growing more tendrils towards each other whenever they fire simultaneously, then that's a memory mechanism, and they can use this to predict in the future that when one of them fires, the other one is sort of.
But in general, so in general, you're capturing regularities in the sensory input.
Correct.
By presuming that if A often fires with B, then if A fires alone, you B also fires.
Yep,
Um,
that's right. That's the Hebbian learning model in a nutshell. And to
okay.
be clear, this isn't like a particularly great or complete model, but it's a very simplified model of like how you start to get basic learning.
Okay.
All right. And with that, we have made it through the like mostly biology section, and we're ready to start talking about minds proper.
Yeah.
Let's pause there for break.
Okay. Sounds good.
Unless we have more questions.
No, I think most of my questions are mindsy.
Go find some more.
Yeah, good.